Welcome back. We are at the last end of module number 6, that is biophotonic technology to detect genetic disorders, and in today's lecture we are going to discuss protein microarray technology. Previously was DNA microarray technology, let us now discuss protein microarray technology. So, these will be the concepts that we are going to cover. Some of these will be covered, not all. So, what is protein microarray technology? Just like DNA microarray technology, the protein microarray is a high throughput technology that immobilizes proteins instead of DNA on a solid surface to study protein interactions and functions on a large scale, enabling simultaneous analysis of thousands of proteins in a single experiment. Proteins are us. We need to understand how protein interact, how protein fold, what are the three-dimensional structure they need to form, and we want to see them happening in a microscopic slide in front of our eyes to understand what could go wrong. DNA based genetic mutations can cause several diseases, cancers and whatnot. Protein based problem with misfolding can cause similar problems such as Alzheimer's or Parkinson's, but here I would like to make a caveat, make a disclaimer that still with those diseases that are associated with protein misfolding, we do not know, like Alzheimer's or Parkinson's, these diseases are associated with misfolded proteins. The three-dimensional structure of the protein that it is supposed to be is not forming properly. Now, because of the disease, the protein has formed, the 3D structure has formed badly, or because the 3D structure has formed badly, the person is suffering from Alzheimer's or Parkinson's disease or dementia, where you are forgetting things, or your motor neuron disease, or your hand is shaking or vibrating all the time. We do not know yet. We see these kinds of misfolded proteins in the patient's body, but we do not know if the proteins have caused the disease or because of the disease the proteins are misfolded. We need to see what causes the protein to misfold and if that has resulted, and we want to see that using this type of array, this type of simultaneous analysis of thousands of protein interaction in a particular temperature, particular pressure, depending on light sensitivity, depending on pH sensitivity, all of those things. So, as I told you, the basic structure is group of amino acids coming together to form chains. They then combine together by hydrogen bond, by ionic interaction, is the local folding patterns of a protein chain. The two most common secondary structure are alpha helices, these kinds of helical, these kinds of round structures, and otherwise there are pleats you may say, like beta sheet, which are stabilized by hydrogen bonding between amino acids. These are the secondary structure. Tertiary structure describe the overall three-dimensional shape of a single protein molecule. It is determined by the interaction between amino acid side chains, hydrogen bond, hydrophobic interaction, electrostatic interaction, and then quaternary structures are also there, where more than one different types of amino acid chains, more than one different types of tertiary structures can combine together. The quaternary structures refer to interaction of these subunits, multiple protein subunits that come together to form a complex. So, if you can consider this as a monomer, then monomer 1, monomer 2 and monomer 3 have all combined together to form a quaternary structure. So, this is the basic structure of protein. So, the fundamental difference between a DNA and a protein microarray is that the DNA molecules are very robust, which can be dried and rehydrated to restore their functions, but no such luck in protein, are highly unstable and easily denatured in solid, liquid and liquid air interfaces. DNA microarray involves hybridization of single stranded or messenger RNA with complementary strand, which is very specific. A will only match with T, C will only match with G. No other reaction is possible. On the other hand, the current antibody production capability result in the production of low affinity current antibody production is very costly. Antibody, though it is very specific, but very costly, and it is pretty easy to make the antibody which you are synthesizing wrong. The chemical reaction, the synthesis of DNA is more or less very easy, ATCG, how wrong you can go, but the antibody that you are producing, you could go wrong, thereby reliability and accuracy is a result. DNA microarray technology can utilize PCR, polymeric chain reaction. Nowadays, every single person knows who has ever done a COVID test or wanted to do a COVID test or know someone from COVID test. Polymeric chain reaction, the DNA or the RNA is simply multiplied, amplified. Protein cannot be amplified or multiplied. A protein is formed, it is formed. Complementary strands are highly specific for their targeted molecule. Antibodies, being generally glycosylated, have large surface area for interaction, and it can have significant cross relativity between target proteins. DNA microarray provides information about gene expression patterns. Protein microarray provides information about proteins, including protein-protein interaction, binding specificity, and what modification takes place. So, there are several challenges in here. Protein microarray technology is still going on, we are still developing, it is still not as robust as we have here in DNA microarray technologies. The surface chemistry has to be improved, appropriate surface chemistry technology has to be done. Proteins at the end of the day are unstable. You take out protein, you put it into a microslide, you put it in top of a slide, a glass slide, and then you do not maintain the appropriate temperature, etc., it will simply denature and die. You cannot return it back. DNA, on the other hand, no problem as such. You keep it in on top of slide and it will survive for a long period of time. Capturing agent selection, detection method, agent selection, identifying and isolating, etu bondo korben. এই জায়গাটা একটু কেটে দেবেন না, আমার অ্যালার্জি হচ্ছে। Let's start. Capturing agent selection, identify and isolate a suitable capturing agent such as an antibody that specifically binds to the protein of interest. Detection method, choose a detection method, usually fluorescence, but chemiluminescence or mass spectroscopy can be employed based on experimental requirements. And then the protein extraction capacity, ensure that the microarray chips allows for the extraction of a detected protein if further analysis is needed. It enables downstream investigation such as protein identification, characterization or functional studies. So, the different stages of protein microarray, you have a surface chemistry, you have a coated slide, polysilene aldehyde, then you have capturing agents like antibodies. We have discussed antibodies before, it is not very specific, but several large libraries are available. Aptamers are small peptide or small RNA molecules, nucleic acids that binds to proteins. Fibronetics and phage display peptides, the more complicated they are, the more accurate they are, the costlier they become. And then the detection method, you have chemiluminescence, fluorescence, sensitive but requires labeling of protein. The problem with fluorescence tagging or labeling is that it can, the fluorophore that is attaching with your DNA or attaching with your protein is probably even changing the property of the protein. Mass spectrometry and surface plasmon resonance. So, mass spectrometry is also possible, but low throughput and very, very costly. How many institute you know that possess a mass spec? A mass spectroscopy equipment is very costly. Surface plasmon resonance, on the other hand, is highly sensitive, and this is the area that I work in, and hopefully in the next chapters I will show you about surface plasmon resonance and plasmonic analysis of these things, but it does not require any leveling. It is also costly, sophisticated, low throughput, but leveling is not required. So, aptamer is something that is coming up very, very strongly. Aptamers are short single strand DNA or RNA molecules that bind to specific target molecules. Here they are not attaching with a specific DNA or RNA, but they are attaching with specific proteins. They are generated through a process called systemic evolution of ligands by exponential enrichment, SELEX, with a large pool of random nucleotide sequence. It is iteratively screened and enriched for binding to the target molecule being your targeted protein. An aptamer can recognize a wide range of targets, including small molecules, proteins, peptides, and even cells. similar to antibodies, but with advantages like smaller size and most importantly unlike antibodies, aptamers are far cheaper, right. Aptamers are small chains of RNA, DNA or peptides that attaches themselves with specific targeted protein molecules, not just proteins, peptides, individual cells, but they can be cheaply produced. They can be cheaply produced, and the ease of chemical synthesis or modification is there, making them even better examples. So, putting protein microarrays, it's also too much of an information. You can read it at your own leisure time. But protein microarrays are produced on glass or silicon substrate that are treated with an aldehyde or similar agents to immobilize the protein's capturing agent, the antibody. We need to surface treat the glass slide with this aldehyde group. The aldehyde group on the substrate reacts with the amino acids of the antibody or protein, forming a Schiff base linkage. This is purely chemistry term to say some sort of a complicated covalent bond. This linkage is responsible for immobilizing. After protein attachment, any remaining unreacted aldehyde groups on the substrates are quenched, washed away. Bovine serum albumin is used in the buffered solution. Bovine serum albumin, the name suggests some sort of a protein, to block any unreacted sites. So, you treat the surface with aldehyde group and the bovine serum albumin. One part connects with the aldehyde group, the other albumin is then made to attach with your targeted molecules. You can print it similarly. A molecular layer of bovine serum albumin is first attached to the glass substrate. The attached BSA is activated using an carbonate. The activation process generates active residues. Any remaining unreactive surface is taken away. This is pure surface chemistry, very little to do with biophotonics, but I have given it just for reference, just for annexure. I will make sure you do not get any questions from this particular topic, because I myself do not understand it. I am not a chemist. And there are other immobilization agent there. I work on surface plasmon chemistry. So, gold is the best thing for me, but maybe the affordability is pretty low, hence bovine serum albumin is used. So, you have an antibody, you then get it attached with the specific material that you are trying to target. Immobilized antibodies have been commonly used. Capturing agents such as aptamers specifically binding to proteins can be used. Photoaptamers utilize light sensitive aptamers. So, the reaction will only happen when you have shine a particular light, and that could be utilized for capturing proteins and reading the array to see if a particular analysis of particular reaction has taken place or not. Oligonucleotides and aptamers often offers the advantage that the same technology used for printing microarrays for mRNA expression can be utilized. So, the detection, this schematic is very easily. So, you have put these kinds of antibodies or aptamers associated with it. Upon either shining light or some kind of chemical reaction, or based on this specificity, they will be captured, and if not, they will not be captured, and then you are focusing light onto this particular area. Either fluorescence based tag molecule, they will shine light or they will modify their presence. Here there is no light, here there is light. So, yeah, the capturing have had actually taken place. From that you can understand that, yeah, this particular protein sequence is present, and or you can what we are trying to do, instead of this fluorescent level associated with it, you simply shine light into each individual spot, try to see the absorption pattern of specific light. The absorption of this will be different than the absorption of this, and combination of this will show a completely different absorption pattern, and thereby without minus the fluorescent level you can actually see if the capturing, if the attachment, the complementary attachment had happened, and thereby you can make some sort of a comment on the presence or absence of a specific proteins on to your sample that you have collected. So, yeah, that is basically the end of chapter number 6. Protein microarrays are valuable tools for high throughput analysis of protein interaction. Proper consideration like surface chemistry and capturing agent selections should be given, and there is an ongoing advancement going on in protein microarray technology, and we will be working on that. I myself am personally involved in making analysis of these kinds of structures. So, go through my references and I will see you in next class. Thank you very much.